How does SPM analyze neuroimaging data? Have you ever wondered how scientists make sense of the complex images produced by brain scans? Today, we will look at how statistical parametric mapping, often referred to as SPM, plays a key role in analyzing neuroimaging data. This method is essential in brain mapping and diagnostics, helping researchers understand brain activity and structure. SPM starts by applying statistical techniques to brain images. It focuses on identifying areas where brain activity or structure shows significant differences across various conditions or groups. This is particularly useful in both functional and structural imaging, such as functional magnetic resonance imaging, positron emission tomography, and single photon emission computed tomography. The first step in SPM is preprocessing the raw neuroimaging data. This involves several important tasks. Spatial realignment corrects for any head movement that may have occurred during the scan. Normalization aligns individual brains to a standard anatomical space, making it easier to compare data from different subjects. Smoothing is another crucial step. It enhances the signal-to-noise ratio by averaging signals over neighboring three-dimensional pixels. These preprocessing steps ensure that the data is ready for meaningful analysis. Once the data is prepared, SPM conducts voxel-wise statistical tests across the entire brain volume. This means that each three-dimensional pixel is evaluated to see if there is a statistically significant difference in brain activity or structure related to the experimental condition or clinical group. The result is a statistical parametric map that highlights areas of significant change. SPM utilizes models based on the general linear model, which allows researchers to incorporate various experimental designs and covariates. For instance, in functional neuroimaging, it can model brain responses to different stimuli or tasks. In structural studies, it can detect differences in brain anatomy between different populations. In the realm of neurotechnology and brain mapping, SPM's ability to localize and quantify brain function supports diagnostics and research into neurological disorders. For example, it can help identify abnormal brain activity patterns in conditions such as traumatic brain injury or psychiatric disorders by comparing patient scans to those of healthy individuals. Practically, the statistical maps generated by SPM can guide clinicians and researchers in pinpointing target brain regions for further study or intervention. This can include neuromodulation therapies or surgical planning. Furthermore, SPM's integration with other neuroimaging modalities enhances our understanding of brain function and dysfunction. In summary, SPM analyzes neuroimaging data by preprocessing images to a common framework, applying voxel-wise statistical models to detect significant brain changes, and producing maps that reveal functional or structural brain differences relevant to brain mapping and diagnostics.